Okay, so my name's Lachlan Curran, and I'm a PhD student at the University of Queensland. So my current work is looking at how land use up in North Queensland changes the function and diversity of the soil microbial community. But at the conference this year, I was presenting my honours research, and that was looking at a deep dive into the drivers of the soil fungal community down at Mary Cairncross in sunny southeast Queensland, and the factors that might impact their diversity and functioning in regards to land use. So the reason I was interested in this project was because soil fungi is an extremely diverse community. In a single teaspoon of soil, you can have billions and billions of individual species. Also, not individuals, thousands of species, billions of individuals. They make up normally the dominant biomass of the microbial community in our woody areas, in our rainforests. So bacteria are important too, but in our rainforests and subtropical areas, fungi are the most important ones. They serve a lot of really, really important functional roles. So we have our saprotrophs, which are sort of the recyclers of our ecosystem. They break down all those difficult chemicals that bacteria and other organisms can't quite access, and they release nutrients for plants. We also have our mycorrhizal fungi that partner with the majority of our plant species, both our endo and ectos. And again, they help plants facilitate nutrient acquisition and responses to stress and pathogens. And of course, there are also fungal pathogens that in a natural balanced system are important for population control, but in degraded areas, they tend to increase in abundance and they then can become quite problematic. So in my work, I just looked at one little case study at a subtropical rainforest remnant up in Mary Cairncross Nature Reserve, about 2 hours drive from Brisbane. And so this reserve is quite interesting because the Sunshine Coast Council over the last few years has purchased different parcels of land directly surrounding the reserve of different land uses. So some of it's quite degraded pasture, some of it has surviving secondary vegetation, and some of it the council has actually started doing active restoration and seedling planting in that site. So I really wanted to know, on this mosaic of land work, how is the soil fungal community responding? The main question being, is the diversity and functioning of the system that you find in the rainforest interior, is that going to be similar to secondary species and pasture species, or is there a change? And the reason this matters is that with land use changes, you have not only changes to your above ground plant and animal communities, but you're changing the soil chemical and physical properties, which leads to big differences in the composition of species and functions of soil fungi. And so this leads to a challenge in restoration where your soil fungal community may not be in a desirable state for restoration. So globally and within Australia, a lot of our plant species that we put in the ground require specific fungal partners and compositions in order to acquire nutrients, survive stresses like droughts, floods and fires, and just grow and survive in competition. And when that changes, they can really, really struggle. So particularly in the southeast Queensland context, a lot of what we put in the ground from seedlings and seed stocks fails to survive long term. And part of that we're revealing is the result of these changed fungal communities. So the two main results that I found is I sampled along all these different land uses, so from within the remnant community, within degraded pasture, and within secondary passively regenerating vegetation. And we found that despite these differences in plant communities, the further out you go from the remnant edge, so the further you go from your existing rainforest, the fewest fungal species you have. Furthermore, in your really herbaceous areas, so in your pastures or things dominated by forbs and other mixes of really herbaceous species, the functional composition is quite low. And what these two things do together means that you're more likely in these areas to have fewer species and fewer functional diversity. So you're going to have the probability of having beneficial fungal species in there that are helping our plants grow, helping our restoration activities be successful is lower, both the further you go away and when you're planting in these degraded pastures. So with that in mind, the two recommendations my work gives to bush care regenerators is plant as close as you can to the edge of the rainforest remnant, so that maximises your species richness, so you're getting more diversity and more potential functional variation. And before you plant, if you're planting in fields or pastures, real grassy areas, add substrate diversity. So add your big leaves and twigs and litter. The best solution is to just take it from your existing remnant and apply it, and just leave that there for a bit. Leave it there for a few weeks before you plant, and that will encourage other functional characteristics in your community to emerge. Like right now we just don't have enough information about specific fungal taxa to be able to say, you know, we want this species and this is how we're going to grow it here. That research is ongoing, it's part of what we're doing. But in order to just maximize the probability of that variation and maximize all the benefits, just by increasing the variation, we increase the number of functions, and by increasing the number of functions, we can increase the stability and resilience within the ecosystem and suppress the overabundance of things like pathogens and non-cooperative fungal species. So yeah, that's about my work in a nutshell.